Welcome to this webinar titled Making Sense Out of Multiplex, When and How to Use the Visiopharm Phenotyping Tools. Today, we're going to cover a number of things at a very high level. Firstly, a brief introduction to what is phenotyping, how it can be useful, and when people do it. I'll then touch on some manual approaches that can be taken in order to do multiplex phenotyping. Next, I'll introduce the Visiopharm approach, which is an easy, unbiased one. And then lastly, we'll touch on some of the ways your data can be explored. We're going to start with an introduction to multiplex phenotyping and ask, what is phenotyping itself? Phenotyping is simply the ability to characterize individual cells based upon the presence or the absence of particular markers. In this cartoon example, I've taken three markers, green, magenta, and blue, which you can see on the schematics of cells on the left-hand side. And in this case, you can see that the presence or absence of these three markers indicates that a cell is either cell type A, where it's solely represented by the green marker, cell type B, which is present for both the green and the blue marker, and cell type C, which is present for magenta and blue. An important concept here is that not only is it relevant whether or not a marker is present, but also that a marker is absent. Cell type C, for instance, may be classified as a different marker if it's a triple positive. Now, this schematic example may seem trivial, but you can actually directly relate cell types A, B, and C to relevant biological phenotypes. In this case, the green marker is CD4, the blue is CD8, and the magenta could be FOXP3. And in this case, we have three different biologically relevant phenotypes of T helper cells, regulatory T cells, and activated cytotoxic T cells. The question becomes, how do we even classify cells as being positive or negative for an individual marker? And in this case, what we do is to simply look within individual channels of our low, high, or ultraplex phenotyping data. This is one example from a fluorescence experiment, and we're just showing the DAPI or nuclear marker and FOXP3 in blue and yellow respectively. In this case, the first step is to define our phenotypic unit by identifying the individual cells. You can see in this case that we've identified all of the different cells, some of which are positive and some of which are negative. We can then use a simple threshold classifier in order to identify cells that are positive for any individual marker, in this case the FOXP3, and separate these cells or subset them. At this point, for instance, counting the number of cells, looking at the distance apart from each other, or looking at other characteristics. Now, this is fairly trivial with a single marker, but what about the example we had before? In this case, three different markers, the green, magenta, and pink and blue from the first example. If we extend this concept of a manual approach to phenotyping, we can look at the same example with the three markers. And what we can do is to start to build up a decision tree for these simple T-cell markers. In this case, first looking, for instance, at CD4 and deciding, is that positive or is it negative? If we look at one example, a triple positive in this case, it's quite a rare phenotype, but you can take a CD4 positive, CD8 positive, and FOXP3 positive cell, and we can form an activated double positive T cell. So this is very simple for one example, but it's important to realize that even with three markers, the decision tree becomes quite complicated. In this case, for three markers, we have 7 choices, and that can produce a maximum of 8 different phenotypes. You can see those listed over here on the right, and this decision tree can be walked through for each different marker to identify positivity or negativity in this case. So hopefully you can appreciate even for 3 markers, this is quite complicated. You can imagine for 6, 7, 10, 15, 20 or 30 markers that are becoming more and more commonplace, these sorts of decision trees become rather ungainly. And this leads me to highlight the problems, or at least two of the problems, with manual phenotyping or this threshold-based approach. First, as I mentioned, it's not scalable beyond a few channels. Even with 6 or 7 channels, it becomes very difficult to build a decision tree to identify each of the different phenotypes. The second problem, which is quite an issue in realistic or real-world data, is that it forces the user to pick a threshold intensity to define positivity. So in this case, the user would have to go through however many channels you have and identify whether or not a cell is positive or negative and define a cutoff. There are many problems with this approach, not least of all that in real tissue, the level of background intensity often changes, the level of autofluorescence or the noise profile may also change, and so picking a single threshold simply does not provide robust results. What Visiopharm have done to address this problem, or both of these problems, is actually create a multiplex phenotyping tool that's fully integrated into the Visiopharm platform. And this is an automated, streamlined, and unbiased approach to multiplex phenotyping. We can take a practically unlimited number of channels to support all modalities. And these include some of the things I've mentioned before, like immunofluorescence and imaging mass cytometry, all within the same platform. One of the key points of the Visiopharm software is that it's actually acquisition agnostic. So in this case, for instance, no matter where the data come from, everything from what we would call single or dual pass immunofluorescence that can have 3 to 8 channels, cyclic immunofluorescent techniques where markers are stained on tissue, imaged, and then stripped and restained for different probes, or imaging mass cytometry techniques, which are doing above 30 markers at a time and continuing to increase the number. All of these different modalities, despite being different file formats and coming from different systems, can all be opened and analyzed using the Visiopharm platform. So let's take a look at how multiplex phenotyping works in Visiopharm. As with other modules, we build up workflows based on modular apps or units. And so the first app would be to do a cell detection or a detection of phenotypic units. And in this case, we lean on our advanced AI deep learning tools in order to do a robust cell detection, even in complex tissue. To take just a second, we always use AI deep learning tools to do cell detection, chiefly because it can cope with the wide variance that we often see. This could be variance in cell size, cell shape, or cell intensity. It also provides a more robust solution to highly clustered environments where cells can be much more easily separated. Moving on to the multiplex, once we've detected a cell outlines, we then simply use our easy-to-use interface here so that the user can detect which channels they would like to include or exclude from their clustering analysis. Very often, we would want to exclude structural markers or obviously the nuclear marker that was used as a cell outline, but sometimes functional markers may also want to be excluded from this phenotypic step, although they can always be accessed later. So the user selects which channels they'd like to exclude from the list of single or multiple channels here, and then simply trains the classifier to automatically find the represented classes in your training data. Some of the parameters here are tunable if desired, and I'll mention this in the next couple of slides. But what the classifier will produce is a list of all the represented phenotypes that are found in the sample data. I'd like to just jump into a little more detail about what happens when the user pushes this train button. All of what I'm about to show you happens in the background, but it's important to understand how much of a time saver it is in terms of defining the positivity and negativity of cells within a population. For this demonstration, I'm going to go back to my original example of a 3-channel CD4, CD8 FOXP3 markers. And here you can see a combination of cells in our samples that we're going to use for training. So when the user provides these cells for training and then hits that train button, the first step is to assess the range of intensity in each channel. And what this basically means is to first start, for instance, with the CD4 channel and look for each individual cell that's used for training. What's the positivity in the CD4 channel? This allows the software to then build up an intensity distribution and an understanding of the range of intensities within the CD4 channel. This is then repeated for a second channel, in this case the FOXP3, and again the intensity distribution is built up into a model. Lastly, we can do the same for our CD8 channel, and again build up an intensity distribution. The second step of training, once we understand or have built a probabilistic model about the positivity and negativity in our sample, is to report the represented combinations. So in this case, we go back not only to look at individual channels, but to understand what's happening on the whole cell level. So in this case, these intensity distributions are used to build a machine learning model. And then we take each of the individual cells in the training dataset and then feed it into this model. So in this case, we're taking a FOXP3 and a CD8 positive cell, running it against our machine learning model, and it will decide using a probabilistic approach that this cell is positive for FOXP3 and CD8. There's one other filtering step to remove very niche classes or classes that are not highly represented, and again, this is user tunable. But assuming this filtering step is passed, then a class is defined as a CD8 FOXP3 positive cell. This is repeated again for other cells in the sampling dataset, and in this case, for instance, this cell is passed through the machine learning protocol, filtered, and then a new class, which is CD4 alone, is added to the list. This goes on and on through the sampling dataset, and then eventually this class list is reported to the end user. This is a really beneficial approach because it saves the user time during development and also builds a more robust model against deciding which cells are positive for which different markers. I will just make one more point when it comes to the training, is that you may have noticed these boxes in one of the previous slides. When we're defining positivity or negativity, sometimes if we have particularly complex data with abnormal noise profiles, for instance, or multimodal intensity distributions, we may wish to refine the data before it's added into the model. And in this case, what the user can do is provide either value ranges or quantile ranges into the classifier. What this does is to take that intensity distribution and actually limit it to a certain part. This, for instance, can cut off low band noise or reduce problems with bleed through from different channels. And at this point, only these data that lie within the thresholds or within the quantiles are used for characterization. So what I've covered in the last couple of slides is how we go about training a classifier. Once this is done at the beginning of an experiment, what we end up having is an app that can be run against more data, either single images or batch processed across a whole data set. And in this case, you can see an app up in the ribbon for multiplex phenotyping. In order to run this back across the image, we simply take an image that has had the cell outlines identified, the same as before, run that app from the ribbon, and what we get is each one of the individual cell masks classified into one of the identified phenotypes. At this point, you can see that all of the masks are color-coded based on their phenotypes for easy identification. And as I mentioned, this process, once the classifier is trained, can be run as part of a workflow. So for instance, downstream of artifact exclusion or tumor identification, and can be run as part of a batch process as well, scaling it up to many hundreds of images. So I'd like to take the last part of this talk to talk about exploring your data. Once we have classified this individual image to identify all the different phenotypic classes within it and have produced these overlay masks, there are lots of different things that we can do with this dataset. For instance, we can produce semi-quantitative or qualitative heat maps. These are great for identifying hotspots of particular phenotypes or combinations of phenotypes. I say semi-quantitative because we can also take these heat maps and use them to define regions of interest. For instance, identifying the hotspots of CD68 macrophage markers, T-cell markers, or any combination, and then further identifying or analyzing these areas, counting, for instance, other cell types. Secondarily, we have a wide range of outputs and quantitative outputs available in Visiopharm, so almost any parameter can be measured and reported on a per cell level as a granular output to be able to identify, for instance, cell size, cell shape, the intensity distribution in that particular class, or even spatial measurements such as the object distance between any two classes, for instance, T cells and macrophages, or any classes and any structure, if you're interested in looking at immune cell infiltration, or the distance to blood vessels, or the distance to tumor boundaries. All of these can be reported at a very granular level as well. I also want to mention that built into the Visiopharm software, we can produce a number of different phenographic outputs. I'm going to touch on these in the next slide. The first is the phenotypic profile, and what this allows you to do is to take the output of a multiplex phenotyping analysis and then plot the prevalence of each of the different classes you see across multiple regions of interest or multiple images. For instance, if you're trying to discern responders versus non-responders or immune activation in different areas, this is a very nice way to quickly be able to identify particular images or particular ROIs that have a response. The second phenograph we can produce are a number of different plots, such as the t-SNE plot. In this case, this clusters similar objects as nearby points and dissimilar objects as distant points. I'll also mention at this point that the colors in the t-SNE plot you see here in the background are also directly related to the colors in the overlays that I showed in the last slide. So it's very easy to take a particular class and identify the cell types back in the original image. Not only can we produce the type plot, as you see in the background, but the per channel heat maps as well to identify the contributions of particular types of cells. Another plot we can create is what we call the phenotypic matrix. In this case, this is very useful for probing the composition of individual phenotypic classes. A good example, for instance, is if you have activated T cells that are positive for CD8 and then granzyme B to make them cytotoxic. In this case, we may want to see in different conditions which cells are more positive or which images show more positivity in the granzyme B class than other ones. Again, the matrix allows you to probe these sorts of questions. The last phenotypic plot I'd like to mention today is the neighborhood analysis. And what this allows you to do, again, very easily at the push of a button, is to run a statistical analysis or a statistical spatial analysis to indicate whether or not the interactions between two different phenotypes are pretty much by chance, or if they're more likely than you would expect if all of the different cells were distributed randomly. This is great for identifying particular cell populations and whether or not they're associating with each other or being actively excluding from each other. Very important in things like antigen-presenting cells that rely on cell-cell interactions. As I briefly mentioned, all of these phenographs are available from the ribbon at the top of the Visiopharm software. It requires no coding or no scripting in order to produce any of these plots or run any of the analysis I've spoken about today. So it's a really easy way for new users to be able to do quite complex exploratory analysis without much training and without much need to get to know new features of the software. The other important key with the Visiopharm software is that your data are always accessible to you. So for instance, if you run a clustering analysis or a phenotypic analysis, you can always export any of the data, the cell positions, the intensities, the classification, size, shape, texture parameters. All of these data can be easily exported for single images or whole data sets into simple text files such as tab-separated value lists. But we can also export the overlays that are produced. So for instance, if you wanted to do a custom visualization, we can always export the overlays as TIFF files, or if you prefer vectors, XML. Just to summarize briefly what we've covered today, the Visiopharm multiplex phenotyping is an easy to use, easy to configure, unbiased multiplex clustering tool. We've taken incredible complexity here and made it very simple to use. At no step do you require any programming, learning new syntactic languages, or coding. We're also format agnostic, so everything I've shown you today can be applied to almost any data type, be it imaging mass cytometry, fluorescence, or other techniques. Importantly, multiplex phenotyping in Visiopharm integrates with existing tools. This means you can leverage the power of our AI deep learning, both for cell detection, but also, as I briefly mentioned, identifying areas of interest, excluding artifacts or necrotic areas, or any other upstream tool that could benefit from AI deep learning. And lastly, the outputs are fully customizable. You really do get to decide what questions you ask of your data. This could be quantitative measures, as I mentioned, as summary statistics or granular per cell data. Density heat maps are very easy to produce and to use, or our built-in phenotyping plots, including t-SNEs and neighborhood analysis. I'd like to thank you for watching this webinar today. I hope you've gained some valuable insights into multiplexing and how the Visiopharm platform approaches these sorts of analyses.